Hi, my name is Wang. I'm a PhD student from Dr. Mike Beer's lab. I'm excited to present my project on the Keystone Symposium of Higher Order Chromatin Architecture, and my title is A Consensus CTCF Loop Map Reviews Global Constraint of Enhancer-Promoter Interactions. So it's been abundantly clear that the 3D chromatin architecture is not randomly organized in the human genome, and structural units like compartments, tags, or loops are identified by experiments like HiC and ChiaPad. So one of the most prominent features of those structural units is that their boundaries are usually bound by CTCF. So here I want to answer 3 questions about CTCF. That is, what determines the interaction specificity between CTCFs, and how similar are CTCF loops across cell types? And finally, does the CTCF loops constrain enhancer-promoter interactions? And for the first question, I just want to notice that the convergent CTCF motif orientation is not sufficient to solve it because there's a huge number of non-interacting convergent CTCF motif pairs that are close to each other. So to answer the first question, we developed a mathematical model of loop extrusion. Loop extrusion is a biological process that's been proposed to explain the formation of CTCF loops. And what we did here is to transform it into a mathematical model by modeling the probability of CTCF binding, the contact frequency decay with distance due to cohesion falling, random cohesion falling when it's translocating along the chromosome. And the third is the competition between a new CTCF loop and an existing preformed CTCF loop. And the fourth one is the CTCF motif orientation dependent loop stability. So by taking the product of all the 4 terms, we obtain the final probability of CTCF interaction. So we applied this model on CTCF ChiaPad data, and we found that this model is capable of predicting CTCF interaction at high accuracy. We obtained AOPRC about 0.6 at a very high positive to negative ratio, and our predicted probability is also highly correlated with the PET count from ChiaPad experiment. To give a concrete example, as we can see from here, our prediction is very similar to either HiC and ChiaPad data from both the loop arc plot and the heat map plot. One of the highlights from our model is that because loop competition is highly correlated with distance, we performed a controlled testing by control one of them and using the other to predict CTCF interaction. And by that, we showed that loop competition instead of distance is more informative and more likely to be the causal factor that really drives the specificity of CTCF loop interaction. So moving on from that, we obtained more CTCF ChiaPad data from ENCODE that's generated by the Jackson lab. And clustering of all the over 20 samples reveals a huge similarity of CTCF interaction across cell types. And we also showed that the CTCF interaction can be predicted by HiC or ChiaPad from other cell types, as well as our loop extrusion model. So due to this high consistency observed from this data set, we derived a consensus CTCF loop list with over 80K CTCF loops in total. And a lot of times, those CTCF loops are nested, meaning that they're overlapping and intersecting with each other a lot, which makes it difficult to visualize and to understand. Therefore, we also applied a HMM to identify the CTCF contact domains from the consensus CTCF loop. And the idea of the CCD is very similar to TAD. And as you can see, a lot of times, the boundary of CCD is very close to the TAD boundary, which implies that those two concepts are interchangeably in sometimes. So finally, we want to use the CTCF looping information to understand the regulation of enhancer-promoter interactions. So our hypothesis is that EP interactions are more likely to occur within CTCF loops and not to across CTCF loops. We proposed a very simple quantification of the CTCF loop constraint by taking the joint product of the CTCF loops nearby in a given enhancer promoter pair. And we used the CTCF looping information to distinguish interacting and non-interacting enhancer promoter pairs defined from the PAL2 chiropathic experiments. As we can see from here, just as we would expect, when the enhancer promoter pair is contained by a CTCF loop, it is more likely to interact. And when it interferes with a CTCF loop, it's less likely to interact. And by combining the CTCF constraint with cell type specific chromatin accessibility information, the prediction accuracy further increases. And here is a brief summary of my results, and thank you for listening.